So this section, 15, the last section, right? 13 was on the innate, 14 was on acquired, and then these are like issues that we have with our immune system, problems. So we're going to go through, most of this is hypersensitivity. There's 4 types of hypersensitivity. You have type 1, type 2, type 3, and type 4 listed there. So you might need, I might expect you to know, when I give you an example, which one is type 1, which one's type 2. And then they have names after them as well, like type 1 could be referred to as immediate. Allergies fit into this category. Type 2 is antibody mediated, and another name for that is cytotoxicity. When we get to that, I'll talk about that. And then there's this what's called immune complex type 3. Now the first 3 involve antibodies, right? IgE is a type 1, remember that. You know, so you might see questions where you have IgG, IgE. You might see questions, and you need to know, like say, which antibody type is associated with different things. So you know, that's important too, distinguishing that. Type 4, there is no antibody involved in that, right? The T cells are involved. They cause it. So I'll talk about that and give you an example. Now, I think I told you before that on mast cells, there you find, it's actually it has what's called an Fc receptor. It's an Fc receptor that binds only to IgE antibodies. So the IgE antibody sits in that receptor, and then that turns the IgE antibody into a receptor. Right? I explained how B cells have the IgM antibody back then. Now, if you have allergies, then on your mast cells you have antibodies, IgE antibodies that bind allergens. Right? An allergen is an antigen that produces allergies. Right? You've probably heard that you've heard of that before. Pollen is an allergen. If you're allergic to penicillin, I guess you would consider that an allergen. If you're allergic to penicillin, I'll talk about that in a minute, then you make an IgE antibody that sticks to it. But it's a hapten in that sense as well. Now, when the allergen binds to IgE, that causes the mast cell, so you can say it's activated to some extent. And then it has granules, they're called, which are basically big vesicles that release histamine. They also release other mediators. If you, say, take a steroid for allergies, what the steroid does is it prevents mast cells from releasing all of those mediators. It's much more effective than just taking an antihistamine. So that's the theory. Now, to make the IgE antibody, it's no different than, right, a B cell being activated by in this case, an allergen instead of an antigen, but it's the same, they're both the same. This causes allergy. You make, the B cell becomes plasma cells, and then they have to secrete, right, I told you, plasma cells, they have the choice. IgG, IgM, IgE, or IgA, against, all directed against a specific antigen. So if you have the capacity or plasma cells that make IgE, then your IgE eventually ends up on the surface of mast cells in your body. You always have mast cells. If you have allergies, then you have mast cells that have made IgE in response to whatever you're allergic to. And the mast cells, right, the mediators, they wreak havoc. So you're basically, your immune system is attacking, things that aren't really hurting you. It just doesn't know the difference. Okay? Right? What activates the B cell? The ability of the allergen, right? If there's a B cell in your body that has the correct IgM that fits to whatever pollen you're allergic to, then your plasma cells make antibodies to that pollen, and then your mast cells will react when you encounter it. So, right, here's they're showing the IgE eventually finding its way to mast cells, and then when the allergen encounters the IgE on the mast cell, then mast cells release many things. I guess there's 6 altogether. Because a steroid will work much better if it's effective. So if you take Nasocort, then what you're trying to do is, the steroid in the Nasocort should only be affecting your nose, right? I mean, it might get into your body elsewhere, but it's concentrated in your nose, and it's attempting to put the mast cells in your nose to sleep. That's kind of what a steroid does. If you take an antihistamine, then you're trying to block just the effect of one of those mediators, which is histamine. Right? Histamine causes all sorts of problems, but so do the other mediators. So, anaphylaxis or anaphylactic, it's immediate, or anaphylactic is another name for type 1. Now, if you're allergic to pollen mold, you don't have anaphylactic reactions normally to those sorts of things. Right? Say 1 in 1,000 or 1 in 10,000 people have allergies that induce an anaphylactic shock, it's called, and we'll get into that in a minute. So it happens fairly quick once you encounter the pollen. It's immediate, so that's another name given to that. If it's localized, then you normally, it can be, you can have stomach problems with it. It can affect maybe your lungs. Sometimes it's your skin. There's people in my family, they eat strawberries and they break out in hives. Now, how that happens, I have no idea. It doesn't make any sense to me. If you eat something, how that activates mast cells in your skin. I mean, if you rub the strawberry on your skin, I can see that. But so, it's not all that clear always why, I can't explain it all, but maybe an expert could. Now, systemic means, right, it's your whole body. So an anaphylactic reaction that's going to kill you would be a systemic response. A respiratory would be something like asthma, which is sort of systemic. You're kind of getting close to whether is that still systemic or not. I wouldn't ask you a question about that. But definitely anaphylactic shock. Because you dilate your blood vessels and you blood pressure drops, then you would die from that. And if you don't give epinephrine in response, now normally the things that induce anaphylaxis are the, you know, it's not very common. It could be a bee sting, you know, if you know, the first time you're given penicillin, you may not react all that badly to it. And it might be, if you give it orally, it might be a different reaction than if you're giving it an IV penicillin. But they have the ability to act as a hapten. So what a hapten is, right, so let's see, type 1, a hapten's involved. Type 2, there's going to be a hapten involved, and there's going to be a hapten involved in type 4 too. But what they do is if the penicillin, right, say this is a protein, and most of the time, the proteins have to be on cells. Not always. But you have a protein, and then say this is the penicillin molecule. It sticks to the protein. And because of that, it becomes an antigen. It's a complete antigen. If it's not stuck to a protein, then it won't cause problems. But it would be, allergy to penicillin would be type 1, right? So that's hapten's important for penicillin, and it's a type 1 hypersensitivity as well. Now, this is pretty complicated. When you induce asthma, it's mast cells in your lungs that are being activated by something you have maybe are allergic to, and then that's causing all kinds of things. Leukotrienes are released, histamine, serotonin, bradykinin, and prostaglandins. The prostaglandins cause pain, right? We said they're involved in the fever response too. There's many different prostaglandins. So they can dilate blood vessels. But the important thing for asthma, right, is that you constrict the bronchi, which is basically a smooth muscle contractor. So to relieve the contraction of those smooth muscles in your bronchi, you can take a rescue inhaler like albuterol. Albuterol reverses that. But I don't understand how you get albuterol there if it's clogged up, so that's always a problem. But if you were to take a steroid, and sometimes there are inhalers that are a combination of steroid and maybe not albuterol, but something like it, and they work both ways, right? But you want to, if you're taking a steroid, then you're trying to block the process. So you need to normally take it once or twice a day, and it may take 2 or 3 days before it's effective. Right? So you can't use it as a rescue if it's a steroid, which is kind of nice to know, I suppose, if you have asthma, or if your children have asthma, you should know the difference. So there are treatments, and I'm not going to get into the different treatments that are on this. The over-the-counter meds, right? These are Nasacort or things like it, or for your nose, and you're spraying steroid. The other thing to use would be, or let's say for I know my eyes itch all the time, and there are antihistamine eye drops that work pretty quickly, or you can even get, I think, eye drops that have steroids in them too. That might, if that's the only symptom you have, then that might be the best thing. There's no reason to take oral steroids that affect your whole body. Just take them where you're having a problem. It's kind of like steroids that you apply on your skin, right? If you have a rash, no reason to take an oral steroid for that. But normally, if you're in if you're in anaphylactic shock or something, a steroid's probably not a bad thing, but it would have to be an IV, something that acts quick. Now, desensitization is the treatment that's been used for a long time. And the purpose is, right, you have plasma cells making IgE, but you want to induce them to start making IgG, if you can. If you make IgG, then you're hoping to, right, have that antibody stick to the allergen before it reaches the mast cell. It's just hopefully when they inject the allergen in your body to induce this, you don't go into anaphylactic shock, which probably pretty common when they do that. So we talked about type 1, type 2, cytotoxic it's called. We don't use IgE here, right? We're only using you can use both IgG and IgM. That's because both of them activate complement. Complement is activated. Right, so the antigens that those antibodies bind to have to be on a cell. Okay, so and then that cell gets lysed by complement. That's the idea. So the red blood cells are a good example of this. Right, antibody sticks to the red blood cell, and then C1 sticks to the antibody, and then the complement causes the red blood cell to lyse. But we'll give you some other examples. Hemolytic disease of the newborn, and this is what's called drug-induced. It's cytotoxic because the antibodies that stick to whatever cell they stick to, right, that cell is destroyed. That's why you refer to it. It's pretty quick. So these are a diagram of what is the antigen found on red blood cell. Now, if you have type O, you have these two little circles. Now, there's just not one of these on all the red blood cells, right? The antigens are on proteins on the surface, but they're just showing you what has to be stuck to the protein to make these antigens. So if you're type O, you have neither A or B antigen, but you do have an antigen on there. Actually, there's hundreds of them, right? But H antigen is what type O people have, which isn't normally a big problem. There are people that do have problems with it. If you add a sugar to the H antigen, I think it's galactose. The other A is another sugar, I'm not sure, I've forgotten what they were now, but that's how you make a B antigen. Just adding one sugar gives you the complete. Now the antigen is this whole thing, but it's different than just having the H. Or if you put another sugar on the H antigen, you have your type A. And all you need is the enzymes that do that. And now the other issue is these antigens you say they are naturally occurring. That means that you're constantly exposed to them in the foods that you eat. So foods that we eat have these two antigens. That means that we make antibodies against them ever since we could, unless there's a problem. You always have, if you're type O, you always have anti-A antibodies and you always have anti-B antibodies, which you guys have learned before. You probably haven't learned the concept of what's naturally occurring. So these are the antibodies. If you're type AB, because they are self-antigens, you're exposed to them in your food, but you're not going to make antibodies to them, or else you would destroy your own red blood cells. Right? But if you're type O, then they have both. Now, I like to show you this because it'd be nice if we did this in lab. Hopefully someday we will. But the little specks are red blood cells. If they clump like this, they are clumping as a result of an antibody. So anti-RH binds to the RH antigen, and either you have the RH, it's also referred to as the D antigen. If you have the RH antigen, then you're A or B or AB plus or O plus. It determines whether you're plus or minus. All right, that's where you get these pluses or minuses. B plus means when you use the antibody to the RH antigen, your red blood cells have that antigen. That's why the red blood cells clump. Agglutinating means they stick together because of the antibodies cause that. They would also cause the blood cells to lyse eventually. If they're in your body, there might not be any complement in this situation, so that if there's no complement available, it won't happen in this kind of scenario, but in your bloodstream it would. So a transfusion reaction is an example of type 2 cytotoxic, right? You activate complement because antibodies in your blood, in your body, are going to stick to the wrong blood type. Right? If you're a B, type B, then you're going to have anti-A antibodies. That's what these are supposed to be. They don't label them, and probably should. And then they attack the red blood cells that you were transfused with, and you get hemoglobin released from the complement breaking up the red blood cells. And you die from, hemoglobin is toxic to your kidneys, and you die real quick. Now, hemolytic disease of the newborn, it's called, another name for it is erythroblastosis fetalis. This occurs in women when they're pregnant because they produce an antibody that they normally don't. So if the dad is Rh positive, right, the red circles are Rh positive red blood cells. They have that antigen. Mom is Rh negative, so she has the negative sign. That means she doesn't have the Rh antigen. Now, this antigen is not naturally occurring. So if you're Rh negative and don't have it, you're not always making antibodies to it like you do with the A and the B antigen. But mom will, if she's pregnant and has a baby with Rh positive cells, and some of the baby's red blood cells gets in her bloodstream, then her body recognizes them as foreign, and she produces anti-RH. Okay, so anti-RH antibodies stick to the Rh antigen. It's not going to affect mom because she doesn't have the antigen in any of her red blood cells. But because it can pass the placenta, if she has another baby and the baby is Rh positive, then the antibodies that mom made can go through the placenta and it's passed on to the baby, and it would destroy the baby's red blood cells. Now, if you get some of, what you want to do is if this occurs, right, some of the baby's blood, normally it happens at pregnancy, or I mean at birth, right, that something goes wrong and some of the placenta blood from the baby gets into the mom's bloodstream. It doesn't take much probably, you know, I would think, you know, fewer than 100 red blood cells would be enough. But if that occurred during birth, if you gave mom, and what we used to do, if mom was, the first thing, you know, as soon as the baby's born, the cord blood goes to the blood bank, and the blood bank types the baby. If mom is negative, and you already know that because you always do blood work on mom. Normally they even cross-match some blood. They have blood ready for mom. But they, if baby's positive, mom's negative, then we gave, Rogan was ordered, it was like a, what do you call it, a standing order. It went right back to mom and they gave her an injection. Now they kind of give it, I think, during pregnancy for some reason, probably to make money. It's not very common for there to be a problem during the pregnancy, but it could happen. But what you want to do is give mom an injection of the anti-Rh antibody. And that destroys the red blood cells from the baby, if any of the baby's red blood cells gets into mom. See, there's not very many of them are. And that would hopefully prevent mom from making, and it must work pretty good because I think it's not a big problem because of that. And then another example of type 2 is called drug-induced, and that the drugs act as haptens here. Just like I told you, penicillin, that was type 1, had to bind a protein. Now we're saying if a drug sticks, you know, we're not saying which drug does, but some drugs are known to cause this. They're haptens, but when they stick to proteins, then you can, right, make an antibody against the drug that causes the cell that has the drug attached to it to lyse. So they're showing here, they can even, you know, platelets aren't real cells. They're like fragments of cells. But if an antibody sticks to them, then your body will remove the platelet. And, you know, complement, I guess, lyses it is some, but the platelet's probably not a good example of this. But technically that can occur. So there's 3 types to this. If it kills platelets, then you get thrombocytopenic purpura. That means the drug is sticking to, the cell is a platelet that it's sticking to. If the drug is sticking to white blood cells, then you lose white blood cells, and it's called agranulocytosis. White blood cells are being lysed. And then if it's red blood cells that the drug is sticking to, then it can cause hemolytic anemia because you're destroying the red blood cells by activation of complement. So we talked about type 2. Now this is type 3, hypersensitivity. Another name for it is immune complexes. Immune complexes are a combination of antigen. Let's say you get an injection to, like, say for a vaccine could cause this, right? You're injected with antigen, and it's just, you know, proteins in your body, maybe in your arm, and then antibodies. You know, you didn't really need it because you already had antibodies to it, but the antibodies that you have in your body stick to these antigens, and you get immune complex. Right? An immune complex is antigen and antibodies clumping together. You know, like 1 or 2 of them hooking up is not a problem. But if this becomes, you know, 10, 20, 30, 40 antibodies all clumped together, then it can generate things like an Arthus reaction or serum sickness. So arthritis would mean you get an injection, say, in your arm of some antigen, right? And that protein, you already have antibodies for it. So before your body has a chance to get rid of the antigen, right? Because it'll do that, right? It'll process the proteins, break it down, macrophages, dendritic cells get in there and chop it all up, make sure it's not a pathogen. If antibodies start, then you can get cell damage and edema in the area of the site. So that's local. If it actually ends up in your bloodstream, maybe you get an IV injection, like an antitoxin, or you get bitten by a snake. Then you can get what's called serum sickness because they form and they might deposit it. And you never know where they're going to end up in your body. If it's in your bloodstream, it might get stuck in your kidney or in your liver or your lung. It gets to be like a big clot, and it's so big that it starts to stick in places. So this is a depiction of an antibody, right? The Y and the little footballs are the antigens, right? The immune complex. Sometimes, right, they attach to blood vessels. So these are some examples. They can occur in your joints too, or on the skin, right? Eczema, seborrhea, psoriasis, things like that could be caused by immune complex. They can form in your kidney, right? But it's because not only do two antibodies next to one another can activate complement, right? C1 can stick there, and then if this is near a cell, then the complement can destroy that cell. It doesn't know the difference, It's just going to kill whatever's close to it. And then the neutrophils can come in there, generate problems too. So it's a combination, right? They're only showing 4 antibodies here for simplicity purposes, but it could be 20, 30, 40, 50, 100, thousands. And then neutrophils. And then this is the area where the complex is, would be that's where you would have the problem. It could be, I think in here they're trying to show endothelial cell and a blood vessel. So that's a blood vessel, but the blood vessel could be in your kidney, the blood vessel could be in your liver. That's type 3. And then type 4 is referred to as delayed or cell-mediated. Right now, it's cell-mediated, just like when we had cellular immunity. We talked about that. We called it cell-mediated or cellular immunity are caused by T cells. So the T cells are involved in this type of hypersensitivity. But because they are, then it's not immediate, right? It can take a day or two before you see whatever the response is. And a lot of times you see the response in your skin. Contact dermatitis is an example, a PPD skin test, and then I'm going to talk about poison ivy and poison oak. But because the T cells are responding to, I guess you could call it an allergen, but it doesn't, allergens tend to be grouped into this category. This doesn't refer to allergies. This really doesn't refer to allergies. But this one you could, right? If you have, even contact dermatitis, they sometimes don't even use the term allergy for that. But you would say you're allergic to the latex because of the reaction you're having. Now, I'm not sure, there might be some involvement with the IgE, but technically, for test purposes, it's a result of T cells. Now, if you get exposed to what's called a hapten, right, as I said before, there's a hapten involved in type 4, and it's a molecule, a chemical found in poison ivy or poison oak. And it is considered a hapten, but it'll stick to skin proteins, and then it's no longer a hapten. It causes a reaction. It doesn't fit these haptens in a sense, but right, we're referring to this as an allergic reaction in this case. But it's caused by T cells attacking the hapten on the skin. And it's pretty itchy too. And the first time you're exposed, technically you just prime your T cells and make memory cells, and you probably won't see a rash. It's the second, third, fourth time that you generate a rash. And it can take, you're exposed to the poison ivy or poison oak, it can take 2 days before you see the rash, which is another problem. But that's type 4. If you get a PPD skin test, they are injecting a lipoprotein called tuberculin. It's a protein found in Mycobacterium tuberculosis. Now they assume that if you've been exposed to that before, that you'll have a T cell memory cells from that encounter that will react and cause a reaction when they inject this protein in your skin. If you haven't been exposed to TB or Mycobacterium tuberculosis, then you shouldn't generate a big reaction. But it's, one of those, it's not a black and white issue. If you don't have, you could use this as an HIV test too. If you didn't see a reaction from this at all, it could mean you're immunodeficient. So if you see an area like this on somebody, you wouldn't know, you couldn't diagnose them with TB, but people that have had TB, even if it's been years, they'll have a scar on their lung from it. So we went through type 1, type 2, type 3, type 4. I know them by their names. Their names really aren't listed on this. Use the ones I have, like cytotoxic, right? Immune complex, I think that's on there. This is also, type 4 is also delayed or cell-mediated. Know the antibodies, right? This one is T cells, type 4. So if you get somebody else's tissue, if you get someone else's cornea, your body's not going to reject that. And even parts of the brain are protected. It's called privileged site. Your brain has its own little immune system. But there's probably not a lot of T cells that get to the cornea of your eye, right? So there are spots that are, you call them privileged. You could get someone else's brain, I suppose. An autograft means you getting your own tissue, should be pretty safe. Isograft is from identical twins. Now, an identical twin, what dictates your tissue type? I think I explained, right? It's the HLA or MHC1. It's actually, there's 4 proteins, right? There might be 10 or 20 different protein 1s, 10 or 20 different protein 2s, 10 or 20, right there. That generates a social security number or a tissue type. Identical twin ought to have identical HLA. But, you know, my siblings, it's not going to be the same. But it'll be pretty close. And an allograft just means you're getting it from another person. If you get someone else's tissue and it doesn't have the same MHC as yours, you want to get as close as you can. You're going to mount a graft versus host response, right, which is the rejection. And you know, if you're not, you've got to take immunodeficiency drugs for the rest of your life, right? Prograft is one that's, I think, is commonly used today. It used to be cyclosporin. Now, if you needed someone like a bone marrow transplant, then hematopoietic stem cells from umbilical cord blood could, if they got some from you when you were a kid, might help. So the HLA or MHC1, I don't have one there, but it's the one. They are right. They give you, say, your tissue type. So you want, if you're, when they tissue type, for instance, you have to have the same blood type, right? Because the A and B antigens are found on cells on your body other than your red blood cells. If you're given if you give someone the wrong blood type, then you have the antibodies to it, they're going to attack that. There's no way of probably stopping that, right? So it's complicated. There's a lot of other things you have to consider. One of them is the MHC1, but that's usually the one that you think of. And then radiation, which is used, but cyclosporin, prograft are drugs that you would take. If you get someone else's heart, they check, usually it used to be, I don't know if it's changed, but every 6 months they would biopsy your heart. If they found T lymphocytes, too many of them, then they would assume your body's starting to reject it. And they would look for another heart. It doesn't always work. Now, right, I used the hapten and 1, 2, 3, 4. Now we're going to add autoimmune. Problems. So we'll give you some examples of autoimmunity and whether you would consider that type of autoimmunity 1, 2, 3, or 4. It's kind of a big leap. Now, what autoimmunity means, it can be you generate antibodies that are found to antigens that are self-antigens. Because we usually leave the term foreign, if I can spell it, foreign antigens. We leave that term foreign out of the description. Right? We just say antigens. But it's inferred. If they're not foreign antigens, they are self-antigens. That means they're not foreign to you. If your body directs a B or T cell response to a self-antigen, then you have an autoimmune disorder. So it just depends on what's being attacked. There are some people that their immune system attacks the pancreas, like beta cells. They get insulin-dependent diabetes mellitus. Right? Some people, they generate antibodies against a protein, or it's basically a receptor on their thyroid, and that attacks their thyroid gland. Okay. Rheumatoid arthritis, for the most part, is generated by immune complexes that are forming inside whatever joint is inflamed. Okay. So you would think of Graves' disease as an autoimmune of type 2 type, right? Lupus and rheumatoid arthritis as type 3 autoimmune disease. And then type 4, they're thinking maybe multiple sclerosis. The problem with these two, when you call it type 4, because you know that T cells are causing the problem, it could be also that the T cells there are attacking a virus. So sometimes, multiple sclerosis is described as being caused by virus, and it might be both. That's the other issue. So it's not always clear. If you have a disease, there could be 3 or 4 things causing it, right? It's not always a result from one virus. It might be 2 viruses, right? Hepatitis is caused by 4 or 5 different viruses. I think there's a D and an E. Now, in myasthenia gravis, the antibodies that are, or they call it autoantibody, attacks a receptor, right? Now, if this is a muscle, and on the muscle you have receptors and the nerves that are basically controlling whether your muscles contract or not. And the reason your muscle contracts is because the neurons secretes acetylcholine. You have acetylcholine receptors on some of your muscle cells. When the acetylcholine hits those receptors, then the muscle contracts, right? It's more complicated than that, but that's pretty straightforward. That's what they're trying to show. If you generate an antibody against this receptor, and some people do, in myasthenia gravis people do, that blocks the acetylcholine. So they get paralysis. So this is a good example, like this autoimmunity stuff, a good example of how important it is that your body, right, it only goes after foreign antigens, or things can really screw you up. Now, immunodeficiency, these are disorders of the immune system we're going through now. Right? Your immune system, for whatever reason, isn't attacking either B or T cell foreign antigens. Okay? It can be congenital or acquired. So something like DiGeorge syndrome, I think it's severe combined immunodeficiency is what SCID stands for. Or this Wiskott-Aldrich syndrome. If you don't produce B and T cells, right, you don't make, I think I was explaining before, you need to have approximately, it's probably over 100,000 different B cells and different T cells if you have a good immune system. If you don't make these, if you lack all those, then you have a congenital, it can be congenital, meaning, right, you're born with it. And it might mean, like if you don't have a thymus and the lymphocytes can't get there, then you can't make T cells, or if they remove it for whatever reason. But sometimes they're just not made, and that would generate a problem. Or if you get HIV, then, right, the virus is actually attacking your white blood cells. Basically, it's the T cells. But if you don't have enough T cells, T helper cells is what HIV goes after. The T helper is needed for T cell activation and B cell activation both. Because I just scratched the surface when I described activation of these cells. It's it's much more complicated. Right, so T helper is what HIV attacks. It actually sticks to the CD4 receptor on T helpers. And then if you don't have enough T helper, people with an active case of HIV, right, they're going to end up with more likely to have cancers, bacteria, viral, and fungal infections. They get the AIDS-like illnesses, I think it's what you refer to. There's a list of about 10 or 20 of them. Kaposi sarcoma. A B cell becomes a B cell in the bone marrow, right? The bone marrow is the primary lymphoid organ for B. For T cells, the thymus is the primary lymphoid organ. That's where they mature. T cells mature because now they make the T cell receptor antigen specific. The B cell makes IgM as the receptor, right? And those, when you say there's 100,000, when I say there's 100,000 different T cells, that means they have 100,000 different T cell receptors. 100,000 different B cells have 100,000 different IgM receptors. So the T cell receptor can bind to 100,000 different antigens. The IgM receptor on B cells can bind to about 100,000 different antigens, right? That's what's the most remarkable thing about our immune system, is that it can attack so many different shapes, and it's stored in our DNA. But that's just here's a little wrap up of T helper activation, right? T cytotoxic activation, and then here's the B cell activation. It's kind of a figure for all three. Does anybody have any questions about that?